Welcome to the deep dive.
Great to be here.
Today we're diving into, well, maybe the biggest question there is, how did life actually get started on Earth?
And not just that, but also why it took billions of years for complex life, like us, to show up, after all that time with just simple cells.
It's a huge story, right? And we've got really interesting stack of sources to help us explore it.
Yeah, we've been looking at papers from Frontiers in Astronomy and Space Sciences, some really key ideas from Nick Lane's work.
Oh yeah, his stuff on bioenergetics and LECA.
Exactly. That, plus a paper on LUCA from PubMed Central, and some discussions around his book, The Vital Question. Lots to unpack.
Okay, so our mission for this deep dive, let's try to get a handle on the environment where life might have kicked off, what that first ancestor, LECA, might have actually been like.
And crucially, how energy, just the sheer amount of available energy, might have controlled the whole path towards complexity.
Right. Expect some maybe surprising connections here. It definitely goes beyond the simple primordial soup idea.
Oh, absolutely. Let's start there, actually. The soup idea, warm ponds, lightning.
The classic picture.
Yeah, but the sources we looked at really point out some big problems, especially, like you said, with the energy side of things.
How so? What's the energy problem with the soup?
Well, think about it. A soup, even if it has all the right molecules, tends to be, well, mixed, homogeneous.
Okay.
Life needs a constant flow of energy. It runs on gradients, differences, like a battery needs a positive and negative terminal. A well-mixed soup doesn't really have those sustained differences, those gradients, to drive the continuous reactions life needs. It's too close to equilibrium.
Got it. So you need a constant energy push, not just ingredients sitting there. So if not a soup, then what?
This is where things get interesting. The sources really zero in on a specific environment, alkaline hydrothermal vents.
Not the super hot, acidic black smokers.
No, a different kind entirely.
Yeah.
Think more like the Lost City system, discovered back in 2000. These are warm, maybe 40 to 75 degrees Celsius, and crucially, they're alkaline, basic, pH around 9 or even higher.
Okay, warm and alkaline, but where's the energy coming from if it's not scorching volcanic heat?
It's geochemistry, a process called serpentinization. Basically, it's seawater reacting with minerals in the ocean crust, like olivine.
Ah.
And this reaction, it naturally produces heat, but also loads of hydrogen gas, H2 and methane, and the reaction itself releases energy. It's exergonic.
And these vents aren't just chemical reactors, they also have a physical structure that's, well, kind of convenient.
Incredibly convenient. They form these mineral towers, but they're riddled with tiny interconnected pores, like a natural sponge made of rock. These pores are microscopic, with really thin walls between them, natural little compartments.
Wow, okay, so you've got tiny rock compartments bathed in warm alkaline water full of hydrogen gas from below. Where's the gradient? Where's the battery?
Between the inside and the outside, the vent fluid inside the pores is alkaline, pH 911, but the early ocean outside. The sources suggest it was probably mildly acidic, maybe pH 5 or 6. Much more CO2 in the atmosphere back then, dissolving in the water. So you have this sharp difference in acidity and proton concentration across those thin mineral walls of the pores.
That's the natural proton gradient, like a voltage difference.
Exactly. That's a great analogy. A natural proton motive force, chemists call it. A drive of protons to move from the acidic ocean side into the alkaline vent side. Plus, you have redox gradients, two differences in electron availability.
And that difference can power chemistry.
That's the idea. It can drive the reduction of CO2 from the ocean water using the H2 from the vent fluid right across those mineral walls.
Which act as
Catalysts, especially iron sulfide minerals like maconite, which are common in these vent structures. They can help shuttle the electrons.
and make basic organic molecules.
Yeah, simple stuff at first, like formaldehyde, formic acid, but it doesn't stop there. The sources also mention thermal convection within these pores.
Like little currents.
Right. Warm water rising, cooler water sinking. This constant cycling could concentrate any organic molecules that form, including things like nucleotides, the building blocks of RNA and DNA.
So they don't just drift away.
Exactly. They get trapped and concentrated. Some lab simulations mentioned in the sources show you can get huge accumulations of nucleotides this way.
Okay, that paints a very different picture. It's not a passive soup. It's like a dynamic chemical engine. Provides energy, materials, compartments, catalysts. and concentration.
It's a much more compelling setup from a bioenergetic perspective. Now, obviously, life still needed those building blocks in the first place.
Right, the carbon, hydrogen, nitrogen, oxygen, sulfur, phosphorus, CHNOPS. How do we get from simple gases like CO2 and N2 to complex stuff?
Well, even before they reached a vent, where could they have come from? The sources touch on a few possibilities. One is space.
Meteorites.
Yeah, we find amino acids, sugars,
Mm-hmm.
even nucleobases,
Yeah.
the letters of DNA and RNA in meteorites that land on Earth today. So some ingredients might have been delivered.
Extraterrestrial starter kit. That's pretty cool.
It is. Or they could have formed here, maybe in the atmosphere, triggered by impacts or lightning. Think hydrogen cyanide, which can lead to other organics. Or maybe locally, near volcanoes, or perhaps even in the vents themselves before life really got going. The key is they needed to get concentrated somewhere with an energy source.
Okay, so we've got a potential cradle these vents, providing energy and maybe helping make or concentrate the building blocks. Let's move on to LUCA.
Right. The last universal common ancestor. The organism from which all known life, bacteria, archaea, eukaryotes like us, eventually evolved. It's kind of the starting point for biology as we know it, for Darwinian evolution taking hold.
So how do we figure out what LUCA was like? It lived billions of years ago, left no fossils.
It's tough, like trying to reconstruct an ancient ancestor just by looking at all its very diverse descendants today. Traditionally, scientists look for things. All cells share ribosomes for making proteins, the basic genetic code. That tells you LUCA could translate genes into proteins, but not much about its lifestyle.
And there's that problem of genes getting swapped around later, lateral gene transfer.
Exactly. LGT muddies the waters. A gene might be widespread today, not because LUCA had it, but because it got passed around horizontally between different groups much later.
So how do the sources suggest we get around that?
There's a more careful phylogenetic approach. You look for genes found in both bacteria and archaea. That's key, both major domains. But you only count genes whose own evolutionary tree, the gene's history, reflects that ancient split between bacteria and archaea.
Ah, so the gene's history matches the organism's history at that deep level.
Precisely. That filters out a lot of the later LG2 noise. And when you take those genes, the ones likely inherited vertically from LUCA, and combine that with biochemistry and geochemistry, a picture starts to form.
Okay, what does this picture look like? What kind of organism was LUCA?
The evidence strongly points towards LUCA being an autotroph.
Meaning it made its own food from simple stuff.
Yeah, from gases. Fixing CO2, probably using hydrogen gas as the fuel. It wasn't eating other organisms or complex molecules floating around. Heterotrophy eating others seems to have come later.
So a gas eater. What chemical pathway did it use?
Almost certainly the acetyl-CoA pathway, also called the Wood-Yondahl pathway. It's chemically simpler than photosynthesis, for instance. It directly uses H2 to reduce CO2.
And that fits the vent environment.
Perfectly. H2 is abundant in those vents. The pathway involves metals, often iron and nickel, in its enzymes, fitting the geochemistry. And it involves these high-energy sulfur bonds called thioesters. It's energetically favorable, it releases energy, and it seems, well, ancient.
Okay, making food from gas. How did it power everything else? Like making ATP, the cell's energy currency?
Chemiosmosis, same fundamental process all life uses today, using an ion gradient across a membrane to power an enzyme called ATP synthase, which makes the ATP. And guess what? Genes for parts of ATP synthase are on that list of likely LUCA genes.
But wait, if LUCA was maybe in these vents and maybe had leaky membranes early on, how could it maintain a stable ion gradient to make ATP? Didn't you say gradients are key?
This is where it loops back beautifully to the environment. The idea proposed in the sources is that LUCA might not have needed to make its own proton gradient initially.
It used the natural one.
It tapped into the existing natural proton gradient provided by the vent itself, that pH difference between the alkaline inside and the acidic outside.
Whoa.
That natural gradient across its leaky membrane, or even across the thin mineral walls it lived in, could have directly powered its ATP synthase. The vent flow would constantly refresh the gradient. Pumping ions actively to create a gradient might have been a later evolutionary step, maybe part of what drove bacteria and archaea apart.
So LUCA was literally plugged into the wall socket of the planet's geology.
That's a fantastic way to put it. The genetics point to H2 use, sulfur metabolism, metal dependence. The biochemistry points to the acetyl-CoA pathway. The geology points to these vents. It all converges. LUCA seems rooted in rocks, as Lane puts it.
That is compelling. You've even mentioned the genetic code itself might hold clues.
Yeah, it's a subtle point, but LUCA's transfer RNAs, the molecules that read the genetic code, seem to have had lots of specific chemical modifications. bits of sulfur, metal groups, things like that. These might be like molecular fossils, remnants of the chemically rich, sulfur-heavy environment where the code first originated and stabilized. Maybe the vents.
Okay, so life starts, possibly in these vents. LUCA emerges, and then not much happens, complexity-wise, for billions of years. Just bacteria and archaea doing their thing. Then, relatively late, bam, eukaryotes appear. Cells with a nucleus, mitochondria, all that complex internal machinery. And seemingly just once. Why? The long wait? Why didn't prokaryotes get complex?
This is the puzzle, right? It feels weird. Prokaryotes are incredibly diverse biochemically. They've had billions of years. Why didn't they evolve big, complex shapes, organelles, things like eyes or brains independently?
Yeah, seems like evolution should have stumbled on that multiple times if it could.
This is where the energy argument, particularly from Nick Lane's work, comes in really strongly. It might not have been about a lack of genetic potential, but a fundamental lack of power. Specifically, energy available per gene.
Energy per gene. Okay, break that down.
Okay, so bacteria and simple eukaryotes, protists, can have similar metabolic rates if you measure them per gram of biomass. They burn fuel at roughly similar speeds for their weight.
Right.
But eukaryotes are often vastly bigger, like 15,000 times the volume the sources give, as an example. Now think about a bacterium. Its energy generation happens at its membrane, primarily the outer membrane.
Where it makes that proton gradient.
Exactly. But as a cell gets bigger, its volume increases much faster than its surface area. If a bacterium just scaled up massively, its energy production, tied to surface area, wouldn't keep pace with the needs of its much larger volume, and all the genes needed to run that volume.
So the energy generated per gene would plummet, it'd be spread too thin.
Precisely. The calculations suggest a simple eukaryote has something like thousands of times more energy available per gene than a bacterium of similar volume would have. And you need that energy surplus for everything complex, building internal structures, moving things around, constantly making proteins. It's hugely expensive.
Is there evidence for this limit in real bacteria?
Yeah, there are these giant bacteria like Apelopicium, huge for prokaryote, visible to the naked eye sometimes. But how do they manage? They don't develop complex organelles. Instead, they pack themselves with thousands of copies of their genome.
Wow.
And they keep those copies near the membrane where the energy is made. Or they have a huge central vacuole, basically dead space, pushing the cytoplasm out to the edges. They increase total energy by maximizing membrane area, but the energy available per gene copy stays low, like in a normal small bacterium. They hit an energy ceiling.
So they can get big, but not complex in the eukaryotic way, because each gene is still energy starved.
That seems to be the limitation.
So what broke the ceiling? How did eukaryotes overcome this fundamental energy barrier?
One single incredibly important event: endosymbiosis.
Ah, the merging of cells.
Specifically, the prevailing theory, strongly highlighted in the sources, is that an archaeal host cell engulfed a particular type of bacterium, an alpha proteobacterium. But instead of digesting it,
It kept it.
it kept it. And that bacterium evolved over time into the mitochondria,
The powerhouse of the cell.
the ultimate powerhouse. Because what mitochondria are, effectively, are tiny, highly folded internal membranes specialized for energy production. Suddenly, the shell wasn't limited to its outer membrane for generating that proton gradient and making ATP. It had potentially thousands of these internal power packs.
A massive increase in internal membrane surface area for energy generation.
Exactly, a quantum leap in the cell's energy budget.
And that energy surplus changed everything.
That's the core argument. This flood of new energy per gene allowed the emerging eukaryotic lineage to afford things prokaryotes seemingly couldn't. A large, complex genome housed in a nucleus dynamic internal scaffolding, the cytoskeleton, intricate membrane systems like the ER. It paid for complexity.
So it wasn't just gradual evolution, but this one pivotal, almost accidental merger that unlocked a whole new way of being.
It seems that way. The endosymbiosis itself might have been a bit messy, maybe not even beneficial at first, but once that energy surplus was there, it created a whole new evolutionary landscape. Suddenly, complex features like programmed cell death, sexual reproduction, phagocytosis engulfing other cells became energetically feasible and evolutionarily advantageous in ways they just weren't for prokaryotes.
It really shifts the perspective. The long reign of simple cells wasn't necessarily a lack of evolutionary imagination.
But maybe just a lack of power to realize those more complex designs.
That's quite a connection. And it's important, as you said, these are powerful theories, well supported, but still areas scientists are actively digging into and refining the details.
Absolutely. The precise nature of the host, the timing, the exact environmental context, there's still lots of active debate and research.
Okay, so let's wrap this up. What are the big takeaways from this deep dive?
Well, first, we've kind of moved the needle on where life might have started, away from the passive soup towards these dynamic, energy-rich, alkaline hydrothermal vents.
Driven by geology itself.
Right. And second, we've got a clearer, more functional picture of LUCA. Not just a list of universal genes, but likely an autotroph, living off gases like H2 and CO2, deeply connected to that rocky, geochemically active vent environment, harnessing natural gradients.
And finally, the reason for the long delay before complex eukaryotes emerged.
Seems tightly linked to energy. A fundamental energy barrier that prokaryotes couldn't easily cross, which was smashed open by the endosymbiotic origin of mitochondria, providing the power surge needed for complexity.
It really ties together geology, chemistry, energy, and biology in a profound way. From the planet's own energy flows sparking life, to an internal energy revolution allowing complex life to finally take off.
It really does. Which leads to a pretty big thought.
Okay, let's hear it. A final thought for everyone listening.
If harnessing energy gradients was so fundamental right from the start for life's origin, and if a massive increase in energy availability was the key to unlocking complex forms here on Earth, what does that imply for life elsewhere? If we find life on other planets, should we expect it to follow similar energy rules? Could the type of energy available on an exoplanet dictate the form complexity of life that might evolve there?